Welcome to this session on digital data presentation tools, a crucial topic in research methodology. Imagine spending months collecting data for your zoology project, only to have your audience completely misunderstand your findings. Today, we will explore how specialized software transforms raw, intimidating numbers into clear, compelling visual stories that anyone can understand. To begin, let us think about the challenge of raw data. Imagine you are handed a massive, printed spreadsheet containing 10,000 rows of numbers representing the daily temperature of a city over 30 years. If someone asks you to find the overall warming trend just by staring at those numbers, you would feel completely overwhelmed and lost. However, if you take that exact same data and feed it into a computer program that draws a single rising red line on a screen, the story becomes instantly clear. Digital tools act as translators, taking the cold, hard language of raw numbers and turning it into intuitive pictures. Now that we understand the problem with raw numbers, let us look at the solution. In the academic and scientific world, digital data presentation tools are specialized software applications designed to ingest, process, and visualize your research data. These tools range from basic spreadsheet software, like Microsoft Excel and Google Sheets, to advanced statistical suites, such as Tableau, SPSS, R, and Python libraries, like Matplotlib. Their primary function is to map abstract numerical values onto visual dimensions, such as length, position, color, and area on a digital canvas. By automating this graphical rendering process, these tools eliminate human drawing errors and allow for dynamic exploration of massive datasets. This brings us to an important question. Why do we place so much emphasis on these digital tools in research methodology? The answer lies in biology. The human brain is wired to process visual information tens of thousands of times faster than written text or raw numbers. If a researcher discovers a cure for a disease, but presents the efficacy data in a confusing, unreadable table, the medical community might ignore the finding entirely. Without these tools, modern research would be trapped in a bottleneck of incomprehension, where brilliant discoveries remain hidden behind walls of impenetrable data. They are the bridge to clear communication. Having seen why these tools are essential, let us peek under the hood. Even though we just click a button to create a chart, the digital tool is performing rapid mathematical calculations behind the scenes to figure out exactly how to draw the shapes on your screen. One of the most common visualizations is the pie chart, which represents parts of a whole. To draw this, the software cannot just guess. It must convert raw data frequencies into precise geometric angles. The size of every slice must be strictly proportional to the category's share of the total data. Let us look at the exact formula the software uses to do this. Here is the mathematical engine behind the pie chart. The formula is theta equals F divided by N multiplied by 360 degrees. Conceptually, this calculates what fraction of the total data belongs to a category, and then takes that same fraction of a full circle. Let us break down the symbols. Theta represents the central angle of the pie slice, measured in degrees. The variable F stands for the frequency, which is the raw count of the specific category we want to plot. N represents the total sum of all frequencies across all categories. If the frequency F increases while the total N remains the same, the angle theta will increase, resulting in a wider slice on the screen. Before we move on, let us see this formula in action with a concrete example. Suppose you surveyed 200 university students about their preferred digital presentation tool. 120 prefer Excel, 50 prefer Tableau, and 30 prefer Python. We need to determine the exact angle the software will calculate for the Excel slice. First, we identify our given values. The frequency for Excel is 120, and the total sample size is 200. We substitute these into our formula, dividing 120 by 200, which gives a decimal proportion of 0.6. Multiplying this by 360 degrees yields our final answer of 216 degrees. Physically, this tells the software to draw a massive slice covering more than half the circle. Now that we have successfully calculated angles for a pie chart, we must address another fundamental calculation: data scaling. When drawing bar charts or scatter plots, the software faces a physical constraint. A computer screen has a limited number of pixels, so it cannot draw a bar that is literally 50 centimeters tall. The software must compress or stretch the raw data to fit perfectly within the designated chart area on your monitor. This is commonly done using a technique called min-max normalization. It ensures that every data point is drawn at the exact correct proportional height relative to the others. Let us examine the formula used for this scaling. This formula calculates exactly where a specific data point sits as a percentage of the total range. The formula is x scaled equals x minus x min, all divided by x max minus x min. Let us define these terms. The variable x scaled represents the new transformed value, which will always fall between 0 and 1. The symbol x is the original raw data point we are trying to plot. The variable x min is the absolute lowest value in the entire dataset, while x max is the absolute highest value. The denominator represents the total range of the data. If the raw value x gets closer to the maximum value, the resulting scaled value approaches 1, meaning the digital tool will draw the bar closer to the top of the chart. Let us apply this scaling formula to a biology experiment. Imagine you are plotting the heights of 3 plants: 10 centimeters, 25 centimeters, and 50 centimeters. The software needs to draw a bar chart where the shortest plant is at the bottom and the tallest is at the top. We need to calculate the scaled value for the middle plant, which is 25 centimeters tall. Our target value, x, is 25, the minimum is 10, and the maximum is 50. We substitute these into the numerator and denominator. Subtracting gives us 15 divided by 40. Dividing 15 by 40 results in a final scaled value of 0.375. Physically, this tells the digital presentation tool to draw the bar for the middle plant at exactly 37.5% of the total height of the chart's vertical axis. Having calculated the math behind the scenes, let us look at the complete workflow these tools execute. On the screen, you see a schematic diagram illustrating this process. On the far left, we start with a raw data table containing categories and numerical values. This represents the intimidating spreadsheets we discussed earlier. The blue arrow shows this data being fed into the digital presentation tool, represented by the computer monitor in the center. Inside that computer, the software applies the angle and scaling formulas we just learned. Finally, the arrow on the right leads to the output, a colorful pie chart and a neat bar graph. The diagram perfectly captures how abstract numbers are transformed into clear, engaging visual formats that anyone can instantly understand. We have covered a lot of ground today, so let us summarize the key takeaways. First, Digital data presentation tools are the vital bridge between complex numerical analysis and human understanding. They take raw, intimidating datasets and transform them into clear visual formats. Second, we learned that this transformation is not magic. It relies on underlying mathematical algorithms, such as calculating geometric angles for pie charts and using min-max normalization to scale data for bar graphs. By mastering these tools, you ensure that your zoology research findings are not just mathematically correct, but also practically accessible and persuasive to any audience. Ultimately, a great piece of research is only as powerful as the way it is presented to the world.